Section 6.6, genetic variation. In organisms that reproduce sexually, they're going to have a distinct advantage over asexually reproducing organs. Number one, because they are much more genetically diverse. They have more variation within the species, okay? And this variation allows those members to respond to environmental change differently. Some will be able to survive environmental changes and reproduce. Others won't, and they'll die off. But this is going to give the population, or the species as a whole, a greater advantage in remaining viable and remaining in good numbers, regardless of environmental change. Factors contributing to greater genetic diversity are number one, crossing over. Remember, crossing over happens during prophase 1 of meiosis, where homologous chromosomes will interchange sections of the chromosome, therefore kind of reshuffling the deck, so to speak, and creating greater variation. The second of these is independent assortment, how the chromosomes line up, or how the homologous chromosomes line up during metaphase to, again, create greater variation within the gametes that are forming through meiosis. And the last thing is random fertilization. We have all these different gametes that are formed, genetically different gametes that are formed, and only one sperm is going to hit the one egg, and it's going to create a unique genetic offspring, much or different from both the parents and other members of that species. So those would all be factors contributing to genetic diversity. So, as I mentioned last slide, crossing over happens during prophase 1 of meiosis. Homologous chromosomes are going to exchange genetic material. The homologous chromosomes will kind of wrap around the arms like so, and they'll break off and reattach, forming two slightly different chromosomes. This section of the chromosome came from here, where it crossed over, and this section of the chromosome came from here, where it crossed over, and what it's doing is making unique or new genetic combinations. This is called recombination or recombinant DNA. You get a mixing of alleles. Remember, alleles are the different versions of a trait, and as a result, you get new gene combinations. Okay, here with the letters, you can see this blue one has all uppercase letters on it. This green one has all lowercase letters on it. After crossing over, this side of the blue guy has an uppercase A and a lowercase b because it got B from its homologous pair. On the other side, this lowercase a combines with an uppercase b to form this side of the homologous chromosome, and you get a shuffling of the chromosomes, creating new genetic combinations. Linked genes are a little bit different. These guys are going to stay together during crossing over, and it's because they're very close to one another on the chromosome. Since they're nearby, they're less likely to separate. They'll get inherited together, even in crossing over, and they don't follow the law of independent assortment, where they're going to sort independently one another. They would be assorted together. Like here, this gene 2 and gene 3, since they're very close together, would probably stay together during crossing over. Gene 4 and gene 1 probably would not, because they're further apart on the chromosome, and it would be easier to break this thing off and reattach it and reshuffle the genes with the homologous chromosome. Independent assortment is how homologous chromosomes can line up in one of two ways for each set of chromosomes. Okay, say for example we have this homologous pair like this, right? That one would be from mom. Okay, and these guys would be from dad. So we have two different things. When they're lined up like this, they could go this way. So when they separated, these two would go that way, and these two would go that way, right? During meiosis 1, and you'd get separated. But they could also line up like this, where we had mom's here and mom's here, and dad's here and dad's here. When these guys separate, we're getting a different combination, right? We're getting different stuff. Okay. Lastly, they could go this way, and this way, and again, when they separate, we're going to get different combinations of chromosomes, and it's random for each pair. What one maternal chromosome does as composed to the next maternal chromosome has no effect on the others lineup, which gives us, in the humans, because we have 23 pairs, 2 to the 23rd possibilities for different genetically unique gametes through this law of independent assortment and process of meiosis. So that's a lot of variation. Okay, so here's a much cleaner drawing of what I did, right? If we have, say, maternals are mom and paternals are dad, these are red, depending how they line up, right? They could go either this way or this way, right? And we're going to get different genetic combinations in the offspring. Notice how these are both red, so we're getting different combinations in the gametes that were formed because of how they lined up during metaphase 1. Now imagine with human chromosomes, there's 23 pairs of these, not 2 pairs of these, and there'll be just a lot more variation within the gametes formed. Random fertilization is completely random. Which sperm fertilizes which randomly released egg? So we have 2 to the 23rd possibilities for sperm cells and 2 to the 23rd possibilities for different genetically unique egg cells, giving us about 64 trillion different genetic variations in the offspring, which is just a lot of variation within our species.